AS in A-level biology 9700, component paper 11, October, November 2022. Question number 1 says, the photograph shows the part of the plant cell. Now here, the actual size is given there, that's 5 micrometer. While the scale is mentioned with the 5 micrometer, and we need to measure this scale with the help of our ruler, that's in millimeter. So we shall also use the smaller unit to improve the accuracy and precision. So that's 20 millimeter. Here, the formula for magnification is the size of image divided by actual size. So we're going to convert the image size 20 millimeter into a micrometer by multiplying to 1,000. And this will become 20,000 micrometer. To calculate the magnification, we're going to divide the 20,000 divided by 5 micrometer. So micrometer will cut with a micrometer, and our answer will be 400 times this image had been magnified. So D is our correct option here. Next question is related to the organelles. So which row matches each organelle with its function? So in this question, we're going to proceed with a column by column. For example, the first column, the microtubule, they make cilia and spindle. So they're making cilia and spindle fibers. So that's role in cytoskeleton. So that is correct. They move vesicle within the cell. So different vesicle or cytoplasmic streaming movement is controlled by microtubules. So that's also correct. They move chromatid apart during anaphase. So they are responsible for distribution of chromosome during cell division. So it's also correct. And they form the part of the cytoskeleton. So this cytoskeleton is giving shape to the cell or maintain overall the symmetry or structure of the cell. So they're also playing their role. So all options are correct. In the next column, we shall proceed with the rough endoplasmic reticulum. And these are column of network of the membranes, and the chemical reactions taking place in these networks. They are separated by the membrane to outer environment. So separate some process from the cytoplasm is an correct option. They assemble the amino acid to make protein. Why? Because they contain a ribosome, and which makes their surface rough. So these are rough endoplasmic reticulum responsible to make protein. While the C option, they modify the protein which make to release from the cell. It is the function of Golgi bodies. While it makes triglyceride and phospholipid, this is function of smooth endoplasmic reticulum, not rough endoplasmic reticulum. So here are the C and D option are wrong. So in the next column, we shall proceed only with the A and B to save time. The Golgi body makes lysosome. It is correct. The function of the Golgi apparatus or the Golgi body is to make lysosome. And this contains enzyme for destroying one part of the cell. This is function of lysosome. Lysosome are containing hydrolytic enzyme, which are performing their role intracellular digestions. Now the next option, now here the option A is correct. So next only we shall verify in the fourth column, and then we shall proceed to the next question. For example, centriole assembles the spindle during nuclear division. So they are making mitotic apparatus or spindle fibers during the nuclear division. So there's a function of the centriole. While it also moves the chromosome apart during anaphase. So but here the option A is correct. While in this way, soon after solving, we should proceed to the next question. I can explain the remaining part. For example, the centriole are the part of basal body in the cilia and in flagella, but they are not directly involved in the movement of the cilia and the flagella. So here we have done. We're going to proceed to the next question. Where are the 80 Swedberg ribosomes are present? The Swedberg is a unit of centrifugation. So after centrifugation, how long the particle will take to settle in their position according to their density and their size. So there are two types of ribosome, one large 80 Svedberg, which are present in the cytoplasm of eukaryotic cell only. And the second one are smaller. These are 70 Svedberg, which are present in bacterial cell or mitochondria or in chloroplast. So here the question is about 80 Svedberg ribosome. So it is present in the cytoplasm of eukaryotic cell, while chloroplast, mitochondria, and cytoplasm of prokaryotic cell, they contain 70 Svedberg. So option 2 is our correct option. Which feature, when the present in an organism, enabled to be identified as a prokaryote? So we need to classify the prokaryote on the basis of present characteristics. So here, for example, we have the prokaryotic cell contain single circular DNA without membrane bounded. So cytoplasmic DNA, or if the naked DNA is present in the cytoplasm, so this is character of prokaryotic cell. So A is our correct option. While the cell wall is present in both prokaryote and eukaryotic cell, nucleus is not present in prokaryotic cell. So that's why we cannot use it for classification. While the ribosome is present in both. So we cannot use the ribosome here. So that's why the A is our correct option here. The next question is about what is the function of nucleolus? Now, nucleolus is a dense part which is present in the cytoplasm, in the nucleus of the eukaryotic cell. And this nucleolus is responsible to make RNA, like messenger RNA or transfer ribosomal RNA. So the D is the correct option. They are responsible to make ribosomal RNA. While formation and the breakdown of a nuclear envelope, that's a function of the other organelles like lysosomes are involved in it. Formation of the rough endoplasmic reticulum, that's not related. Synthesis of ribosomal protein, so this is the function of rough endoplasmic reticulum or the ribosomes. So here, the question number 6, the diagram show the result, the number of the tests on the solution of the biochemicals. So these are food tests are there, and in this food test, the Benedict solution is used for the test of reducing sugar, which are all monosaccharide or which are disaccharide like only maltose. So Benedict solution upon heating will give a brick red color. So, but here the blue color remains same. So it means it is negative result. No reducing sugar is present. I'm going to skip the second one because it's not a standard test. While the third one is an iodine test for the starch. And it's giving blue-black color. It's a positive result because the color for iodine is the yellow or the brown, and it will change into the blue-black. So it means the starch is present. If the starch is present, so it means it is non-reducing sugar. So starch is polysaccharide, it's non-reducing sugar, it's also present. While the negative, another test is biuret test for protein. So blue color change into lilac, but it remains same. So it means the protein is absent. So here, our correct option is non-reducing sugar and starch only. Why? Because in B, the protein, and in C, the reducing sugar, and in D, the word only are wrong. We're going to proceed to the question number 7, and it is describing about the molecules. For example, which diagram shows the ring form of beta glucose? So ring form of the beta glucose, so these glucose are hexose sugar. So glucose is an hexose sugar. So all these molecules are showing hexose sugar. So these are the glucose, but the beta glucose have hydroxyl group above the carbon number 1. When you see the option A, so the carbon number 1 contain hydroxyl group above and the hydrogen is below. So this is the beta glucose. Rest of all contain hydroxyl group below. So these are alpha glucose. The next option is about, we need to identify the different type of molecule. So which row correctly matches the example with the type of molecule? For example, disaccharide, it consists of the 2 units, but the fructose is monosaccharide. It's only 1 unit. While the starch is polysaccharide, they contain so many units. So that's why these are two wrong options. So in the next further column, we shall not proceed with this. We're going to proceed with the C and D. Why? Because C, maltose and sucrose are disaccharide. The next, the macromolecule. Macromolecules mean the molecule which are organic and contain a larger molecular mass. These are macromolecules. So ribose, nucleic acid, and cellulose, both are macromolecule. So we're going to proceed to the third column, monomers. The monomer is mean and they contain only single unit. So fructose is a monomer, while the sucrose is a dimer. This is disaccharide, so it contains 2 units. So our correct option is D. We're going to proceed to the fourth column to verify it. The collagen is polymer. So D is our correct option here. Rest of all, in the column number 4, the starch, glycogen, cellulose, all are polymer is correct, but you can find your option in third column correct. So we're going to proceed to the next question. In this type of question, we need to exclude the wrong answer. For example, the enzyme α-amylase hydrolyzes the amylopectin, but it's not able to hydrolyze some of the glycosidic bond. For example, according to the statement, it's only hydrolyze 1,4-glycosidic bond. So when we see that the 1,4-glycosidic bond is present in A, C, and D, there may be an option, while 1,6-glycosidic bond B is excluded. Why? Because it is present between the carbon number 1 and carbon number 6. So that is excluded. While it is not able to hydrolyze the last bond of the chain. So last bond of the chain, last 1,4-glycosidic bond is a D, that is excluded. And option number 3, it is not able to hydrolyze the bond in the certain 3 unit attached by 1,6-glycosidic bond to another chain. So A is a part of the chain. This consists of the 3 units. So that's why it is also excluded according to this statement. So the remaining statement is C. So C is our correct option here. The molecule X is a lipid molecule. Now, this lipid molecule is produced by condensation reactions when the alcohol react with the fatty acid chain, and it will make acylglycerol by formation of ester linkage. Now, this ester linkage, oxygen is coming between alkyl and carboxyl bond. So these are between one side is alkyl group, other is a carboxyl, and this oxygen is making ester bond. Now here, which row is correct for molecule X and triglyceride? I drew the triglyceride beside. This contains 3 fatty acid chain, while 1 glycerol is making with that 3 ester linkage. Here, the statement number A, 1 unsaturated fatty acid chain is present in X molecule, acylglycerol. There's no unsaturated. You can see there is a single bond is present between carbon-carbon atom. So that's why all are saturated. So that's the wrong option. No ester bond, while you can see the ester bond is present between alkyl and carboxyl group. So while in D, 2 fatty acid, in X, so there is only 1 fatty acid chain is present. So C option is correct because only 1 fatty acid chain is there. We're going to verify it into the next column, the triglyceride, 3 fatty acid chain are present. So you can see in a triglyceride 1, 2, 3, 3 fatty acid chain are present. So that's our correct option. The bread contains a mixture of a polypeptide known as gluten. So gluten is a type of a protein which is present on the surface of the bread to entrap the carbonate molecule to produce dough. So 2 of polypeptide found in gluten and glutenin. So this is in the gluten are glutenin and gliadin. So there are 2 polypeptides which are present in the gluten. So that's our glutenin and gliadin. So which statement describes the tertiary structure of the protein? So protein contains 4 different structures: primary, secondary, tertiary, and quaternary structure. So what is the correct definition for the tertiary structure? 3-dimensional folding of 1 polypeptide chain by hydrogen bond or either ionic bond or disulfide linkage or hydrophobic interactions. So that's making tertiary structure. In option A, the disulfide bond formed between the that's glutenin and gliadin. Now, these are two polypeptide. When the two polypeptide join together, they're making quaternary structure. So that's why it is not tertiary structure. So in this way, a large proportion of amino acid in gliadin are glutamine. So if there are the high glutamine are present or low glutamine are present, it is not defining the tertiary structure. So alpha helix sections are found in a glutenin and gliadin. So alpha helical structure is defining secondary structure. The secondary structure contain alpha helical structure and beta plated sheet that's present in that. So that's why this is not tertiary structure. So amino acid with a hydrophobic R group are found on the inside of the glutenin. So these hydrophobic bond are present inside, or hydrophobic interaction is present inside of the tertiary structure, while hydrophilic are present on the surface of the tertiary structure, which is giving its globular shape. So D is our correct option. A student was provided the two test tube, one containing the 10 centimeter cube of P, while another contained the 10 centimeter cube of the Q solution. When these solutions were mixed together for after the 24-hour, the concentration of the P decreased, while the concentration of the Q remains same. So it means the chemical reaction in the P is taking place with the help of Q. Because before mixing, there was no chemical reaction. After mixing, there is a chemical reaction. So it means P and Q are responsible for our chemical reactions. So if the P is decreasing, it means logically it is converting into one form to another form. While the Q remains same, it means the Q is involved in chemical reaction, but at the end of the chemical reaction, it remains same. So there's a role of Q is as catalyst, which is not changing, but involved in chemical reaction, while the P is working as the substrate of the catalyst, which is converting into the product form. So that's why we're going to see that according to this catalytic reaction, which statements are correct. P may break down over the time. Yeah, it's converting into one form to another form. That's why it's decreasing. Q may be biological catalyst. Yeah, it is the biological catalyst because it remains same. P may be a substrate for Q. So that's the options. So 1, 2, 3 are correct option related to catalytic actions. The point number 3, that's a 13. Which statements are correct to the lock and key hypothesis and induced fit hypothesis for enzyme actions? So for the working of enzyme, if the enzyme active site stays same and during the catalytic action, so this is lock and key hypothesis. If the active site of enzyme is changing or modified by the penetration of the substrate into an active site, so in this way, this is the induced fit model. So that's going to modify its shape. So in this way, we're going to see that which statement is correctly explaining this function. So this way, the substrate is the same shape as the active site. So if the substrate is exactly complementary to the active site and it's not changing its shape, so that's a lock and key hypothesis. The substrate is held in the place to the active site by temporary bond. So it's overall the correct statement, like it's making temporary hydrogen bond, but it's not defining either lock and key or either induced fit model. Number 3 statement, the enzymes and sometimes the substrate change shape slightly as the substrate molecule enter into the enzyme. So when the substrate molecule will come into the active site of enzyme, it will slightly changes or modify its shape. So this is defining the induced fit model. So 1 and 3 are correct option here. The next question is related with the enzyme with inhibitor. So if we are using a student investigate the rate of enzyme activity with an increase the substrate concentrations. So the experiment was repeated with the addition of reversible competitive inhibitor. So a graph was plotted to show the results. Now, if you are using non-competitive reversible inhibitor, so this non-competitive inhibitor binds other than active site of enzyme. So it is not affected by actual substrate concentration. So if you're increasing the actual substrate concentration, so inhibition rate of the reaction will stay same. So when you see this concept, so B is our correct option, because by increasing the actual substrate concentration, there's no effect on the enzyme actions. So in this way, there is no direct effect. So this one, the B is our correct option. What about the D, the competitive inhibitor? If you're increasing the substrate concentration, so the normal rate of reaction is being achieved. So this is competitive inhibitor. What about A is a non-logical statement, and C is also a non-logical because inhibited reaction cannot be greater than to a normal reactions. So that's why it is the B is our correct option here. The next question is about for the calculation of the surface area to volume ratio. So a cuboidal epithelium cell has the length of 2 micrometer. This cell is approximately cube shape. What is the most accurate estimate of the surface area to the volume ratio? So now these lengths are given there, like each length is 2 micrometer. So it's cuboidal. So all length, like length, width, and height are with the 2 micrometer. So we can easily find it the surface area. So first we're going to find it the surface area of one side. So length multiplied with 2 multiplied 2 is equal to 4 micrometer. This is the surface area, the 4 micrometer square. So of one side. Now, 4, we're going to multiply with a 6 because this contains a 6 side. This is 24 micrometer square of whole. So what about the volume? So volume will be length multiply width multiply height, and this will become 8. Now, we're going to find the ratio of the surface area to the volume. So in this way, the 24 over 8. So this is going to produce 3:1. So here, the option D is our correct option here. We don't need the unit here because in the ratio, the units are deducted, so we are not using unit with that. Which statement is correct for facilitated diffusion and active transport? So facilitated diffusion is a kind of the selective diffusion, and active transport is also a type of selective diffusion because the particular or a specific molecule can move from across the plasma membrane or selectively permeable plasma membrane. But the difference is the facilitated diffusion don't need energy, while active transport need ATP energy. So the option A is the direction or concentration gradient changes. So that does the direction of movement of molecule. So now it is affected for, it is applicable for facilitated diffusion, but it's not affecting to the active transport because facilitated diffusion depends upon concentration difference, while the active transport is not depending the concentration difference. So that's why it is wrong option. So B, the molecule always move in the same rate as the simple diffusion. So molecule will not move in the same rate because facilitated diffusion depend upon the concentration difference. So when the molecule are moving from high to the low concentration, their concentration difference is decreasing. So the rate will change. So that's why B is the wrong answer. So here the D, the molecule ATP is required to move specific molecule quickly through the protein. So it's only needed for active transport, not for facilitated diffusion. So our C is the correct option. Specific molecule are transported across the membrane because both are type of transport taking place by selectively permeable membrane. So that's why these are specific molecule are being transported. So that's the C is our correct option. The equal size of potato pieces are placed in a test tube and covered with a sucrose solution. Now this test tube was left for 30 minutes. So all other variables were standardized, mean temperature and the concentration of the solution, volume of solution, everything stayed same. After 30 minutes, the potato pieces had not changed in size. If there's no net changes in the size, so it means that the solution is isotonic environment. So no net movement of molecule across the membrane is the same. Why? Because the solution contain same salt concentration. So that's an isotonic environment. So in this way, total number of molecules which are moving from outside to the inside and inside to the outside, they will stay same. So the correct option is what? The D is the correct option. The water potential is same in potato and in the sucrose solution. That's isotonic environment. And there's no net movement of water molecule into and out of the potato. So mean the total number of the water molecule are moving from outside to the inside is equal to the inside to the outside. What about the A is a wrong option is what? No more movement of the molecule is a wrong. While in the B, here saying only molecules are moving into the potato. So if the molecule are moving into, obviously the size should increase. And the C, the same, the wrong is no more. So that's one, it should be no net movement. So the next question, number 18. How many of these processes produce genetically identical type of cell? The cells are produced genetically identical by the type of division, that is mitosis. In the result of asexual reproduction, single cell produce two genetically identical type of cell. So here, growth of plant by vegetative propagation, that's also is taking place by mitosis. That's this is included. A replacement of dead cell, repair of damaged cell, asexual reproduction, all are the type of division are mitosis. So that's why all four are correct option. The next question, which row shows the correct number of each component of single chromosome at the end of a prophase of mitosis? So at the end of the prophase of mitosis, condensation reaction is almost completed and the chromosomes are properly visible. So we can identify these chromosomes under the light microscope and we can see their structure. So in this way, these chromosomes are in paired. They contain pair of chromatid. So you can see in the left side in this diagram, so you can see they contain one centromere. So the two chromatid are connected with the one centromere. Each chromatid contain one DNA molecule. So in each DNA molecule, there are two strands of the nucleotides. So it means the total there are 4 nucleotides. There are 2 chromatid, 4 nucleotide strands, and the telomere mean each tip contain a repeated unit of guanine or cytosine to prevent the loss of gene. So that is the telomeres are 4 because 4 tips are present. So that's why the B option is correct here. The which process occurs during the interphase? So the cell contains the interphase or M phase. So during M phase, the cell is dividing, while in interphase, the cell is growing and preparing for division. So here, the DNA replication, S phase of the interphase. So this is taking place in S phase of the interphase. What about microtubule organization? That's a part of the prophase of the M phase. So this is not included in interphase. Synthesis of ribosomes. This is taking place in G1 and G2, growth 1 and growth 2 phases. So that's why 1 and 3 options are correct. Now next, the embryonic stem cell are able to replicate continuously. What happened to the telomere during the repeated mitotic cell cycle of embryonic stem cell? So these stem cells are able to divide again and again. Why? Because they contain telomerase enzyme. And this telomerase enzyme can repair the lost part of the telomere again and again. So that's why their telomere remains same. So the D is our correct option. So they stay same. Why? Because telomerase enzyme is constantly repairing it. The rest of other options are wrong. Which nitrogenous bases are pyrimidine? So nitrogenous bases are generally classified into the pyrimidine and the purine. So the purine are it consists of the double ring, like that's our guanine and adenine, while pyrimidine consists of the single ring, that's thymine, cytosine, uracil. So that's our major concept. Now, according to this concept, we can find the C is our correct option because this contains thymine and uracil. These are single ring. What about adenine and option A? B contain guanine and D contain guanine. That's in the part of the purine. The next question is about theoretical point of view. This is explaining the replication of the DNA. So the DNA replication in the bacteria was investigated, and the bacteria were grown in a medium with the only heavy nitrogen, isotope number 15, until all the bacteria DNA was heavy. So we had been growing the bacteria in the medium, which contained the food of nitrogen isotope number 15. So it means all the nitrogen present in the DNA of the bacteria will contain isotope number 15. So their DNA is a heavy. Then we took the one sample of bacteria and we transfer it into an isotope number 14 nitrogen. So another medium, the new medium with the light nucleotide, this will explain the semi-conservative mode of replication. How? For example, if we have the DNA molecule. Now, this DNA molecule contain in first generation, that's parental generation, this contain isotope number 15. This DNA will divide or will split into two single strand. Now, each single strand will form a complementary part from new medium, while new medium contain isotope number 14. So in this way, in generation 1, 100% the DNA will be hybrid. While the percentage of the heavy DNA is going to be a 50%, while the percentage of the light DNA is going to be the 50%. So what about the second generation? Once again, the DNA will split into two strands. Now, in this way, each DNA, the single strand will form a complementary strand. When it will form the complementary strand, so in this way, we can find we are having the two hybrid and two completely light strand. So in this way, in the second generation, 50% is hybrid, 50% are light. While the percentage of the heavy, if you want to find that, so in this way, 2 are heavy strain. What about the 6 are light strain? The total are 8. So if you want to calculate the percentage of the heavy, 2 out of 8, so this will become 2 out of 8, so multiply into the 100, so this will give us 25%. We can see that the question is saying hybrid DNA contain heavy and light DNA. So which row is correct for the second generation? So when we see that in the second generation, the percentage of the heavy DNA strain is 25%, while the percentage of the hybrid DNA molecule, so the 2 are hybrid, 2 are light, so this is going to be 50%. So B is our correct answer. Now, this next question is the sequence shows the series of the bases at the start of the gene. So if there's the gene is going to start and these the sequence of bases are given there, and messenger RNA and transfer RNA, they are playing their role to arranging the amino acid according to the sequence of DNA. Now, this DNA sequence is given there, but we shall arrange according to the key, which is mentioned here. So the key is giving like amino acid, like alanine, it should be 2. The glycine should be 3. Methionine is 1, and valine is 1. So these triplet codon are there. So we need to see that which triplet codon is coming in one time. So you can see TAC is coming one time. And the second one is valine, CAA is coming one time. So we're going to put MET, methionine first. Why? Because when we see that the option, so all options are representing methionine is coming first. So that's why we're going to put first as the methionine. Now, after that, we're going to see that the CGA is repeating how many times? It's repeating 2 times. 2 times is coming CGA. It means it's confirmed it is alanine. So we're going to put this alanine and alanine. So first and last, second and last. What about the CCA? It's a glycine. It is repeating 3 times. So it confirmed it is glycine because it's coming in 3 times. So we're going to put the glycine, glycine, and glycine. And in this way, our sequence will complete. We're going to compare this sequence and which option contain exactly similar type of a sequence. So we're going to see that the B is completely matching with this sequence. So this is our correct option. Now the table shows the mode of actions of the 2 antibacterial drugs that can affect the synthesis of the protein. So these antibacterial drugs are mode of actions are given there, and rifampicin is one, and streptomycin is another. Rifampicin binds to the RNA polymerase enzyme, and it stops transcriptions of messenger RNA, while the streptomycin is cause error in the translations. So in this way, logically, if we are applying these type of antibiotics, so some messenger RNA are already synthesized in the cytoplasm. So these messenger RNA will be subjected for the translation, and the protein will produce, but that will be abnormal protein. So in this way, the option number 1, the transcription will stop with the help of the rifampicin, but the faulty protein may continue to be synthesized. That's the correct option. Now, if the translation has started, protein may be faulty, it's also correct. But the translation will be inhibited, it's wrong option. Why? Because the streptomycin is only making the error in the translations. So that's why it is the wrong option. So our correct option is 1 and 2 only. The next question is about the bifacial leaf. The electron micrograph show a cross sections of the leaf when and some of neighboring cell. So which structure contribute to mass flow by the use of proton pump? So this mass flow method of the flow of photosynthetic product in the phloem. So we need to know the structure of the leaf. Now here on the above is epidermis, below is palisade mesophyll, and in the central midrib is a vascular bundle in the vein. So in the vascular bundle, above is the xylem always, and below is the phloem. So mean the phloem is a part where the proton pumps is taken place, while below is a lower part, is a lower epidermis part is there. Now here, when we see the option A is a xylem, so in this way, only there's a transport of water and mineral, there's no active transport. While in the D, this is lower epidermis, this is responsible for protection or transmission of light. It's not involved in the transport. While the B is not directly related, it's not part of the vascular bundle, while the C is the phloem, which transport the sucrose and amino acid by the mass flow method. And it's also pump the hydrogen from inside to the outside by companion cell, so by the active transport. So that's why the C is our correct option. Here, you need to know about the position of the xylem and phloem. For example, in dicot root, the xylem is present in the center star shape, while the phloem is present in between the arm of xylem. While in the dicot stem, the above is the phloem and below is the xylem always. While in the dicot leaf or bifacial leaf, so you can see above is the xylem and below is the phloem. While in the dicot stem, outside will be phloem and inside is a xylem. So according to these numbers, when we see that, our correct option is A, because number 1 is the phloem, number 2 is the phloem, number 3 is the phloem in the leaf. So what about the number 4 is xylem in the leaf? So here we can move to the question number 28. Which description of the xylem vessel element is correct? So we need to know about the characteristic of xylem. Xylem vessels, a different cell connect end to end, and the central wall is disappear, and it's making long hollow vessel. by joining the cell end to end. And there's no cytoplasm because these are dead cells. And there's a lignified cell wall to prevent the loss of water. And the pits are present between the cell to provide adjacent transport. So here, which description of the xylem vessel element is correct? So the description mean A, the cell joined to form a tube and the pit at interval, the sieve plate. Word sieve plate is placed in only in phloem, so that's why it's the wrong option. What about the B, that's containing cytoplasm is a wrong option. While in the D, companion cell is present in phloem is a wrong option. So the correct option is it contains elongated cell with end wall are broken down and located in a vascular bundle in the stem and in the leaf. So these all characteristics are matching with xylem. Which property of the lignin are important for the function of the xylem vessel in the stem of tall plant, such as a tree? Now, this lignin is providing adhesive property with the water. So that's why it is inflexible, so does not bend easily. So the xylem has a two function. One, transport of water and mineral, and second is to provide support to the plant stem. So the tall plant is standing on the base of reinforcement or a steel work that's present in the center of the stem. That is by the xylem. So that's why the lignin, which is present in the xylem, is providing inflexible. It prevents it from bending. So that's why it's correct option. It is not permeable, so it prevents escape of water. It's correct option. It is strong to resist the collapse under the pressure. So it is also strong. It prevents the collapsing, so it keeps it open. So that's also a correct. While it has weaker adhesive forces, so the word weaker is wrong. It has stronger adhesive forces to the water molecule than to cellulose. So our option 1, 2, and 3 are correct option here. We can proceed to the question number 30. Which row is correct for the sucrose concentration and the water potential in a source and in the sink in the potato plant 12 p.m. So the 12 p.m. is a middle of the day, where there's a high light intensity and high temperatures available. In high light intensity, rate of photosynthesis will be higher, while the rate of metabolism will be also higher. Now, in the high rate of photosynthesis, it is producing high sucrose or high the glucose in source, while it will consume the sucrose in sink due to the high metabolism. So it means during the day or during the mid of the day, the source will contain higher sucrose, while the sink will contain lower sucrose. Now, if the sucrose is higher, so it means the water is lower. So water potential will be lower. So in the column, in the next column, it will be opposite of this. For example, the source contains higher sucrose, so the water potential will be lower. And the sink contains lower sucrose, so the water will be higher. So in this way, the option B is our correct option. So we're going to proceed to the next question here. The 31, the diagrams show one possible way in which sucrose may be loaded into a sieve tube element. So how the sucrose is loaded. So the first is companion cell are given, and with the adjacent to the companion cell, there is a sieve tube elements are present. These companion cell, they pump the hydrogen ion outside to the neighboring cell wall to the source cell by active transport. So the ATP is used in active transport in number 1. While when the hydrogen ion concentration increase outside, they start moving from outside to the inside by facilitated diffusion. So in this way, the sucrose also move by facilitated diffusion as in co-transport. So number 2 is a facilitated diffusion. While if there is a high sucrose is present in the companion cell, it will transfer to the neighboring sieve tube element. Is number 3 is facilitated diffusion. So the option number 1 is correct. Option C is correct here. So which statement about the arteries are correct? So arteries can resist to the high pressure. So yeah, the arteries are thick wall and there's a high collagen fiber. They have the flexibility so they can withstand with the high pressure of the systolic part of the heart. So now high systolic pressure of the pressure. So it's the artery can withstand with this. So what is the correct option? What about the artery pump the blood out of the heart? So it's not a function of the artery, it is a function of the cardiac muscle, so or a heart to pump the blood. Now the blood in the artery have same flow rate as in the vein. Now the flow rate is a different. Why? Because the blood is moving in the artery by the heart pressure, while in the vein there's no heart pressure. So that's why the word same is wrong here. The pulse is in the artery is a result of surge in the blood that caused the expansion of the arteries wall. So this is the correct option. Why? Because we can feel the pulse in the arteries. Why? Due to the systolic pressure of the heart. And there are semilunar wall at the junction of arteries and in the heart. So that is the arteries when there is a semilunar wall is present, so to prevent the backflow of the blood. So, for example, if there's arteries which are connected with the pulmonary artery, so or which is connected to the heart, so in this way there's a semilunar wall is present in their junctions to prevent the backflow. So in this way, the 1, 4, and 5 are correct option here. The question number 33, what happened during the ventricular systole in a mammalian heart? So ventricular systole is a part of the cardiac cycle. For example, sinoatrial node will give electric supply to the apex part of the heart that contain of right and left atria, which contract and transfer the blood from atrium to the ventricle. And then atrioventricular node will give electric supply to prokaryotic tissue to the bottom of the heart. And this bottom of the heart are the ventricle wall. These ventricle wall will contract and transfer the blood from inside to outside. And in this way, when the ventricle will contract, the pressure in ventricle will be greater as compared to the atrium. Semilunar wall will open, atrioventricular wall will close to transfer the blood from ventricle to the outside. So that's how the concept. We're going to see that which statements are correct. Atrioventricular node transmit an electrical signal to the apex of the heart, so it's basically bottom of the heart because ventricle is present in the opposite to the apex, bottom of the heart. Now the pressure in the ventricle drop below to the pressure in atria. So it's basically the pressure in ventricle, if there's a ventricular systole, so the pressure in ventricle will increase above the pressure in atria. So that's why its opposite word is written. It's the wrong statement. And atrioventricular wall close and the semilunar wall open. So that's correct statement. These are the correct statement. So number 3 is our correct statement. The D is our correct answer. A shortage of protein in the diet of the children can lead to a disease that cause excess of the tissue fluid to build up in the abdomen. So whenever some extra tissue fluid is accumulating in the body part or abdomen, like this is kind of edema, kind of the condition in which the extra tissue fluid store in the body and it's not moving back. So what explain why the buildup of excess tissue fluid can occur in this disease? So what is the main cause for that? The shortage of the plasma protein decrease in blood water potential. So the plasma protein is basically solute, while the water is a solvent. So if the solute is decreasing, so the solvent should increase. So that's why it is the opposite is written, like a shortage of plasma protein, there is an increase in the blood water potential. So that's why the B is our correct option. The shortage of the plasma protein increase the blood water potential. And less tissue fluid will return. Why? Because if there's a blood water potential is already high, so the water will move back. So in this way, the B is our correct option. Now, D is wrong because it's also saying that decrease, it's also wrong. While the C is overall the correct statement, because if there is a low supply of the protein, This will cause the weak cardiac muscle. But this weak cardiac muscle should decrease the water in the tissue. Why? Because due to the low hydrostatic pressure or low blood pressure should transfer low quantity of the water into the tissue. Because if there is a low pressure of the blood, low water should move into the tissue. It should not accumulate. So that's why it is an opposite statement. So that's our, you can say, irrelevant statement. So in this way, the B is our correct answer here. Now, the red blood cell may contain a molecule known as 2,3-biphosphoglycerate. So when this 2,3-biphosphoglycerate binds with the hemoglobin, a higher partial pressure of oxygen is needed to bring 50% saturation of the hemoglobin with oxygen. So it means like 2,3-biphosphoglycerate is inhibiting or the decreasing the affinity of hemoglobin with oxygen. So that's why it need more substrate to get 50% saturation. So in this way, the curve in a Bohr shift, it will move toward right by the presence of 2,3-biphosphoglycerate. So we're going to see that which option is correct. The 2,3-biphosphoglycerate in the red blood cell cause the oxygen dissociation curve shift to the right. Yes, it is shifting to the right. You can see the red is shifted in the form of black. It's moving to the right. So it needs more oxygen to get 50% saturation. So that's why it is a correct statement. The binding to the 2,3-biphosphoglycerate to the hemoglobin reduce the Bohr impact. So it's basically a transfer the curve to the right, and it increasing the Bohr shift. It increasing the Bohr effect, not reducing. So that's the reducing is the wrong word. So the binding of 2,3-biphosphoglycerate to the hemoglobin lower the affinity of the hemoglobin for oxygen. 3 is the correct statement. What about 4? When the 2,3-biphospho is not present. So this will be a normal condition. For example, if the inhibitor is not present, so there will be a normal condition. So whenever the oxyhemoglobin reach to the body tissue, so there is a high concentration of the carbon dioxide, low partial pressure of oxygen, loading will be high. But here it's written less. So this is the wrong word. So our correct option is 1 and 3. So the B is our correct option here. We're going to proceed to the next question here. The carbon dioxide is continuously produced by respiring cell. So when we see that, the carbon dioxide will enter into the red blood cell, and it will combine with the water in the presence of the carbonic acid, and it will produce the carbon dioxide will combine with the water in the presence of carbonic anhydrase, and it will produce carbonic acid, mean H2CO3. This carbonic acid is an unstable molecule, and that will split into a hydrogen ion and hydrogen carbonate. Now, this hydrogen carbonate will move from inside to outside of the red blood cell, while the hydrogen ion will stay inside of the red blood cell. Now here, if the hydrogen carbonate ion are moving from inside to the outside, this will disbalance the charges. So in this way, there is a buffering effect, or this is kind of the effect in which a chloride shift effect. This chloride shift will transfer the chloride ion from outside to inside. As like buffering, how it's working, but it's not buffering. It's balancing the positive and negative charge in the blood plasma. So in this way, it causes, so the correct option is what? It maintains the balance of the positive and negative charges between the cell and in the plasma. So what's going to happen in the blood plasma near the body respiring cells? So the carbon dioxide, when it will enter, so the hydrogen carbonate is moving from inside to the outside, chlorine is moving from outside to the inside. While the opposite is taking place when this red blood cell will reach to the lungs. So it's not related with the pH value, so it's not balancing the pH. So it's wrong statement. It's not affecting to the Bohr shift, or it's not affecting to the affinity of hemoglobin with oxygen or carbon dioxide. So that's why the C and D options are wrong here. So here we have done. Now the question number 37. So the photograph show the sections through the bronchus. So first, the part of the respiratory breathing trach is the trachea, and second is the bronchus. Now, this bronchus, which annotated label is correct? So there the different labels are given there. For example, the B is cartilage. So what about here? Elastic tissue stretches when the breathe in. So this not stretches, it stays same while it keep the breathing trach remain open. So this is cartilage which keeps the bronchus open and prevent from collapsing. So that's wrong. While the C is that's a gland. So this gland produce and release the mucus, while the D is the columnar epithelial cell sweep the mucus toward the throat. So it's also keep the mucus away from the throat and from the breathing trach. So in this way, these are columnar epithelium cell, not squamous epithelium cell. That's wrong. So A is the correct option. These are smooth muscle changes the diameter of airway. So they are placed below the epidermis. So this one, and these are the smooth muscles, and they are the changing or modifying the airway. So that's the correct option, A. Now, the second last question, the penicillium is an antibiotic which used to treat the infections caused by bacteria. This penicillium is a type of bactericidal antibiotic because it's killing bacteria. So it's basically, penicillium is a type of inhibitor of enzyme, which is responsible to make the cell wall of the bacteria. So if the bacteria is growing in the environment where there's a high penicillium is present, so bacteria is unable to maintain normal, strong cell wall. So if there's a weak cell wall, so the water move from outside to the inside and bacteria will get high hydrostatic pressure and it will burst and it will die. But there are some bacteria, they are by chance or by the random mutation, they start producing an allele, and this allele will translate into a protein, and which is making the penicillinase enzyme. This penicillinase enzyme will break down the penicillium. So in this way, the penicillium cannot work on that bacteria. So this bacteria has resistance to the penicillium. Now, which event will explain how the bacteria become resistant to the penicillium? So you can see the events are, number one, random mutation. So this mutation does not depend upon presence or absence of penicillin, and this is a random mutation. Now, this random mutation provide advantageous character to the bacteria to survive or to adapt in the presence of the penicillium new environment. And in this way, the bacteria will survive. If the bacteria survive, this advantageous allele will transfer to the next bacteria by vertical transmissions or bacteria conjugation process. So in this way, this allele will transfer to the next generation. So in this way, we're gonna classify which option is correct here. So the number of bacteria with the allele for resistance to the penicillin increase, yes, because the bacteria, if they have the resistance or resistant gene, so they will survive, so their number will increase. Resistance to the penicillin is the result of mutation in the bacteria. Yeah, so in the result of mutation, the bacteria will produce allele which has a resistance gene. Now, bacteria with allele for resistance to the penicillin pass to the allele to their daughter cell. So it's also a correct option, while the 4 is a wrong option. Why? Because the word always wrong. So the mutation for resistance to the penicillin is always caused by presence of the penicillin, because the mutation is a random process. It does not depend upon presence or absence of the penicillin. You can see it can be accelerated in the presence of the penicillin, but it does not depend upon that. So it is a random process. So that's why the 4 is our wrong option. So our correct option is 1, 2, and 3. So B is our correct option here. This is the second last the question 39. Which statement explain why some vaccines can be taken by the mouth, but the tuberculosis vaccine has to be injected? So this tuberculosis vaccine, they contain antigen, and these antigen stimulate the immunity system to produce antibody. So this antigen is in protein in nature. So the correct relevant the expression which is supporting this statement is the tuberculosis antigen necessary to produce an immune response. So this immune response are what? Antibody, and these antibodies are protein in nature. And this protein will be digested in the stomach and in the small intestine. So if they will be digested, so they won't able to work against the bacteria. So in this way, tuberculosis is injected into the blood. So it should produce the antibody, and this antibody should bind with the bacteria and kill it. What about the option one, like macrophage present antigen in the vaccine to stimulate an immune response? This is not relevant to this because we need the antibody in the blood, not in the stomach. So macrophages are present in the tissue of the body organ, so that's are not relevant. While there are no B lymphocyte and T lymphocyte in the stomach. If there would be B lymphocyte and T lymphocyte in the stomach, they would produce antibody in the stomach, and antibody will be digested. So that's why this is not in the option. So the 2 is the more relevant option, which can easily define it. So that one. We're going to proceed to the last question. A student wrote the following statement about the production of monoclonal antibodies. So which statement is not correct? The monoclonal antibodies are genetically identical type of antibody. So their production process is like following. So first we take a non-self antigen. The antigen which can stimulate our immunity system, these are called non-self antigen. This non-self antigen will stimulate our B lymphocyte to produce plasma cell. Now, this plasma cell combined with the cancerous cell, why? Because it don't have high dividing capability. While it can produce the specific type of antibody, but we want like it should divide again and again. To get these characteristics, we will just fuse this cell with the cancerous cell and we produce hybridoma cell. Now, this hybridoma cell will have the capability to divide by mitosis. It will divide again and again and produce genetically identical monoclonal cell. And these monoclonal cells are able to produce genetically identical monoclonal antibodies. So this is overall the process. We're going to see that which option is correct according to this and the whole process. So first is non-self antigen is injected into a mammal. That's correct. Plasma cell are secrete the specific antibody in response to the non-self antigen. This is also a correct statement. And selected hybridoma cell divide by mitosis to form colon. So that's our correct statement. So what about the wrong statement here? There's supposed to be B lymphocyte or plasma cell fused to the cancer cell to form hybridoma cell. And here's a written T lymphocyte. So the question is, which statement is not correct? So the D is a not correct statement. So with this, we have completed the paper, and hopefully it will be helpful for you. If you find any question is if you did not understand any question, so you can also write in a comment box. I will further individually explain that question for you. Thank you very much.